Can everybody hear me? Yes, okay. Thanks so much for the introduction and for the opportunity to come speak to you today. Just a brief outline for my talk today. It's going to be a little bit different in the sense that I'm really going to cover, given my role in the organization, sort of a broad survey of the different projects that the Allan Institute is undertaking. So I'll give you a brief history and understanding about the Allan Institute and what we're about. Talk briefly about the mouse atlases, and then spend the majority of the talk talking about our adult human brain atlas, our developing human and non-human primate gene expression atlases, again going over how we created those data sets and then a few data vignettes from those. Talk about a new effort that we're launching on connectivity in the mouse, and then spend the last few minutes talking about future directions for the Allan Institute. So the Allan Institute for Brain Science, we were really founded about 10 years ago. Activities didn't get going until 2003, founded by Paul Allen, who's one of the co-founders of Microsoft. We're dedicated to making tools and information readily available to the scientific community, and we're doing this with what we call a biotech-based approach. We're very project-focused, milestone-driven, and we bring together multidisciplinary teams to all make this happen. And again, we do this with this product focus so that everybody is working together. We're all on the same team, and we all share in the successes and failures together. So we have lots of great people that work at the Institute in a variety of different disciplines. 170 staff now we're up to. 40 of these are PhD level folks in a variety of different disciplines, as you can see here, and ever growing. We just acquired additional office space in Fremont so that we could expand some of our labs in our existing building. We now have 50,000 square feet. I should add that Dr. Christoph Cook has recently joined us as our Chief Scientific Officer, and I'll tell you at the end a little bit about what we envisioned for the future with Christoph. What we are not is a traditional PI-driven research organization. There are plenty of wonderful organizations out there. We don't need to be another one, and we are not an extramural funding agency. So the history of the Allan Institute goes back to 2001 with some planning meetings where there was a big discussion around really, I think, big open discussion with lots of luminaries in the field to decide how best to tackle something as hard as understanding consciousness or thought or these very abstract concepts. Fortunately, advisors to Paul Allen really steered Paul to saying, you really need to start here. You need to start from the basics, the building blocks. Over time, you'll eventually get here, but there's so much basic work that needs to be done in this space first. And so that's really where we started. The recommendation was to the mouse genome had just been sequenced, was to see where all of the genes in the mouse genome are turned on in the mouse brain. We did that with our mouse brain atlas, started in 2003, and largely finished that work in 2006. And we subsequently been moving into areas such as defining cell types genetically. I'll tell you about some work there. We're now doing this connectivity atlas, and as I'll tell you later, we're starting to contemplate moving into areas of electrophysiology, neural computation, circuits, as we move forward. So if you go to the launch page of the website, you can see the variety of different projects that we've undertaken over time, and we've been adding to them here. So this is just at brain-map.org, and I'm going to go through many of these projects in a little more detail in the next slides. So this is the Allan Institute as we've been stretched over sort of our first decade of existence. We've added, as I mentioned, the mouse brain atlas. I'm going to talk about some other projects, mouse spinal cord, developing mouse brain atlas. There are other smaller projects that had focus questions that we've also put the data out there for. The things that we're focusing right now on are some follow-up on our developing mouse brain atlas, our mouse projectional connectional atlas, and then a new project that we're launching that I'm not going to talk about today on cell networks. And this is a little bit different, but it's about understanding how cells, individual cells, take information, store it, process it, and towards an understanding there. And our system is going to be neural stem cells differentiating down neuronal pathways. Good. So briefly, we started with gene expression atlases in the mouse brain. Our platform for data generation was in situ hybridization. It's been our workhorse to date, but as you'll see as in my talk later, we're moving much more into more quantitative platforms such as RNA-seq and microarrays. But we've started out with in situ hybridization for its ability to show within individual cells where a gene is turned on, but it's a gene-by-gene approach, and it's inherently non-quantitative. It's qualitative assay at best. So we started out by generating a series of reference atlases that allowed us to create a reference volume. We then register our two-dimensional data that we generated with in situ hybridization, map it, and put it all together in web-based search form. We ultimately created a data set which extracted this information into a data structure that's basically 50,000 voxels by 20,000 genes, which allows a deeper mining of this data. The mouse spinal cord atlas was a project that we completed, I think, in 2006, 2007. This was following the heels of the mouse brain atlas and just leveraged the core platform that we created. So this looked at two different stages of the development, juvenile P4 and adult. We looked at four different sections, CTL and S, throughout the spinal cord. And we covered about 20,000 genes, and all of that data, again, is available. We also moved on to the developing mouse brain atlas, who was sort of covering some of the dimensions that many people had asked for. So the original project was on the adult mouse brain. We realized that in order to do, we had to pick in terms of our dimensions we like to cover. So in this project, we covered about 2,000 genes. There's about 800 transcription factors that comprise that data set, along with other genes that people would find developmentally interesting. But again, this is 10% of the genes available. But we covered multiple stages, so we started at E11.5, 13.5, 15.5, 18.5, and then postnatally P4, P14. And so we create a nice linear structure across time to look at gene expression. We did our final data release on this one, where I say final, but we're always trying to add a few new genes or redo things which we're concerned about the result that we got as compared to other data sets. So those tools are all available, both in 3D and ultimately we hope to do some 4D analysis looking at this data. Okay. So I wanted to move now into our work on the adult human brain atlas. This is a project that we launched around 2008 in terms of moving on to the next step. And we really wrestled with this idea of the concept of an atlas and what people would want. We knew that people really liked in situ hybridization for its ability to show you down at the individual cell level, but getting the amount of human tissue that you would need to do was impractical in terms of thinking about that. So we came up with a project in which we would do basically both aspects of processing, where we would take whole brains and ultimately end up with microarray data on about 1,000 structures, and I'll tell you in a lot more detail about how we do that. But then we're also following that up with selective in situ hybridization with specific genes on specific structures to complement that. And it's just a compromise between not being able to do the full mouse brain atlas type of approach on the human brain. So the process that we've undertaken, we start with a human brain, and I'll walk you through these in more detail in each of the steps, but basically collect DTI, MRI, various block face images. So we go back and forth between a wet lab and anatomy, further annotating, ultimately extracting information, putting on a microarray, and then putting it into our system, and then creating tools for people to view it. So we start out in a medical examiner's office. This is the medical examiner's office in Maryland. We also work with a brain bank at the University of California, Irvine as well. So we work with brain banks on both coasts. And it's actually quite a laborious task to collect human tissue. We make sure that we collect the brain and process it and do all the processing steps that we need to do within 24 hours of time of death. We are actively screening for what they call a psychiatric autopsy, so no history of psychiatric disease. We do a full toxicology workup to make sure that there are no drugs of concern on a board, a whole series of workup. And because of this, our pipeline for obtaining brain tissue is very, very small. Indeed, so small that we've only been able to obtain a handful of brains in this way. And we've really only been able to obtain one female brain using this. And that's just the nature of obtaining high-quality human tissue. We end up failing out most of the brains, even that we collect, that pass our first criteria based on RNA degradation and other things as well. Okay, so once the brain is obtained, we obtain a DTI as well as an MR. This in Maryland is able to be done in cranial. So there's a distinct advantage here in the brains that we collect at Irvine. We have to do it ex cranial. And that's just a logistics issue. So once the brain is obtained, we slab it, freeze it. Those frozen sections are sent to the Allan Institute for Brain Science for further processing. We take those slabs, we do full coronal sectioning, stain them, and then the anatomists come in and make some basic anatomic associations, both in FreeSurfer as well as on these. Those then dictate the structural delineations that people first do to fragment the brain into smaller pieces for us to handle. We then section this, put it on slides, and either do laser capture microdissection, or in the cases of the cortical areas that we obtain, we do a scalpel dissection. So this just shows our macro or our cortical dissection and our sampling strategy here. I'm not going to go into the details of the anatomic assignments, but there's lots of support information on what was selected for the various sampling. But basically, we've taken close to 600 samples for each brain in the neocortex. And ultimately, these come out on microarrays. Here's the LCM workflow, just to highlight here. So for any given structure, for example, the hippocampus, I think this is 63 different samples that we collected through the hippocampus. And when we do laser capture, of course, we're taking multiple sections, so we're sampling in all different dimensions to obtain these samples. But it's still, it's a coarse sampling. But as I said, each brain is partitioned into about 1,000 parts. And when we put this on microarrays, we put this on an adjuvant microarray. Just to talk a little bit about the features of that, this is a custom designed microarray. We're typically sampling with 2 different probes per gene. One was selected by a sort of default adjuvant selection, and then we typically picked another one. Some probes we actually tiled all the way through, and all of that data you'll find on the web. So sometimes if you're looking, you see a whole series of different readouts for a particular gene. It's because we tiled throughout that gene. And these microarrays have all kinds of QC design parameters that are put in there, and I won't go into the details today, but we spike in various metrics to make sure that they perform correctly. We have sample tracking spike-ins as well to make sure that there have been no sample swaps, et cetera. Okay, so let's see here. So everybody has a professional video now, so we had to have our own as well that describes a little bit of the process here. So we put this together. Actually, I gave a TED talk recently, so it was to give some perspective here. But we've collected now data for 2 brains. We're processing more. As I said, there's about 1,000 samples per brain that have been put on microarrays. These all come together in these toolkits that we've created. This is the Brain Explorer. Many of you may have seen it for mouse if you haven't seen it yet for human. And this just highlights some of the features of this, where you can see all the anatomy, you can see all the various subcomponents of the process that we've collected over time, but you can also see the gene expression. This is one particular gene that you can see in heat mapped areas where we've sampled and taken the information. And in addition to the 3D Brain Explorer, we have a whole suite of other tools that enable things like looking at our original coronal structure delineations. You can see exactly where we've drawn, and I think it's an important feature of these kinds of atlases to photo document everything so that you can really make your own determination. If you actually want to see what we're calling this particular region of the hippocampus, you can actually go in and look at the primary data. You can see what we drew, and you can also see the area that was cut out with LCM and make your own determination if you feel comfortable with that assignment and what we collected. Okay? So the kinds of things now that are enabled by doing this, so you can go in, you can look at a whole class of genes from a given pathway. And again, we're sampling broadly the whole genome here. The monoamine pathway, for example, and you can look at them in these sorts of ways where you can see how they're spread out across the brain. Or another thing you can do with these correlative searches is sort of a blast search for a pattern similarity. So finding things that are similar to your gene of interest. And when you do this, you find some surprises and some things that are, things like somewhat undiscovered, like an ORF here. One thing we did on the larger scale was to look at how gene expression allows for analysis of this data on a sort of a larger scale. And this is Mike Harlan is sitting up in the audience. You can query him later on exactly what he did in terms of mathematically to generate this plot. But it's in essence, it's a principal component type of analysis where individual gene expression patterns are all put together. And discriminating patterns are able to be pulled out of that in terms of their overall expression. And what you get out of such an analysis is a recapitulation, even in the neocortex, of the structure of the neocortex purely based on the gene expression values that you get. And we did a first study before we launched onto this larger human brain atlas, where we looked at the neocortex. We looked at 1,000 genes across multiple stages multiple areas of the neocortex in multiple individuals. All of that data has been online for several years now, so we already knew going into that we would find interesting things like this. And indeed, so of those 1,000 genes, we found about 150 that showed regional differences. Here's some striking examples of synaptotagmin, SCN3B, which show different expression levels. This is all within one individual at different Brodmann areas in the brain, BA17, 18, and 21. Same thing here. Okay. So one thing we were very relieved to find, because in launching this project, we knew that the investment that we're making in individual brains is enormous, but we knew that we weren't going to be able to run the numbers up very high in terms of running hundreds, if not thousands of brains. We're really limited to a handful of brains that we could look at. So we were very concerned about the variability that we might see across humans. We were, I think, relieved to find that there's some order here. It's not complete chaos. And indeed, if you take something that's a sort of a top cut of the most variable genes that you can find and look at them in a summary of 150 structures, you get a fairly nice correlation here across individuals. And again, it's just sort of rewarding to know that that when we do this small number of individuals, that people will be able to take things of meaning out of this. Now, there are plenty of differences that certainly are going to be interesting to follow up. And then back to the cortical study, we knew that going in as well, that about 5% of the genes, or 50 out of the 1,000 that we looked at, showed some variability when we looked at multiple individuals for these genes. And here's some examples of these kinds of things. They're typically very subtle. They're within individual cell types or classes within the brain. Here's an example where renin is just turned on in this one versus here, disc one here versus others. And given the variability among individuals, these are all unrelated individuals, we of course don't know if this is due to genetic background or experience or anything of that sort. However, it's a relief to know that the variability is comparatively small. As I mentioned, we're also doing in situ hybridization on the adult human brain, and we're also doing in situ hybridization on the developing human brain. I'll show you in the next part of the talk. But what we've done is now been processing areas in the subcortical forebrain, looking at these sort of macro structures with a collection of genes, again, very small set of genes. We started out looking at excitatory and inhibitory system genes, 60 of them in total, and again, that data is all available on the web, but effectively blocking the brain out into various regions, doing in situ hybridization, again, on a smaller number. This just highlights some of the data that we see here. And again, it's a work in progress. We're continuing to generate this data. Now, moving forward, we're actually now doing what we call a dual processing of our brain. So rather than take the entire adult human brain and process it with using microarrays or in situ hybridization, because we're getting so few good adult human brains through, we're taking one hemisphere and we're processing that with microarrays. We're taking the other hemisphere and we're doing in situ hybridization with it. And that's the process that we'll be doing for the next year and a half or so to generate additional data to start adding to the N in our collection. And brains 1 and 2 are out there and they're available for anybody to use. Brains 3 and 4 are qualified. They're in pipeline right now and being processed. Okay. So I'm going to move on to talk about some work that we've been doing in gene expression in the developing human and non-human primate brain. Much of this work is actually funded by the NIH in various ways. We'll highlight here. The first one being the NIH Blueprint. This was a project that we launched several years ago to look at postnatal development in the macaque monkey. This was a sampling at 0, 3, 12, and 48 months, again with a small number of in situ hybridization, a small number of genes, but key genes per structure. They're slightly different genes depending upon the structure, but nonetheless it's a very valuable data set to see because you get this over time, and again limited to a series of different structures here. And this work has been ongoing. In addition to adding to this, we've been doing laser capture microdissection atlases, and we're going to be continuing this work to add to this in prenatal stages as well. But the kind of data that you get out of this is rather nice. So again, we create these laser capture microdissection atlases in the monkey. We can do now comparisons to the human. We can also look at in situ hybridization and see a variety of different genes. So things we discover here, sometimes we feed in and do follow-up in situ hybridization. So there's a project that was funded by National Institute of Mental Health, and so kudos to Tom Insel for deciding that he really wanted to take the stimulus dollars for NIMH and apply them to a large-scale resource such as this. The BrainSpan project emerged out of that. It's a collaboration between Yale University, USC, different groups there, and the Allan Institute. The Allan Institute fits in that we are generating our cellular level in situ hybridization. We're doing the data access and the sort of the housing of the data and the tools. The anatomy reference framework There's a huge component of transcriptional profiling using RNA-seq that's being done at Yale and USC, and this work is just now completing, and a lot of that data is actually out there. So they've been doing both RNA-seq as well as exon array data, looking at SNPs, methylation, a whole bunch of things. And so just to go over the scope of that project, and some of this data is already available, and more will be coming out shortly. On the data generation side, there's transcriptome, genome, and epigenome analysis. So in the breadth of coverage for these projects, there's about 1,000 samples that are being processed using RNA-seq, exon array analysis, et cetera. So it's a pretty massive effort for RNA-seq. There are 13 developmental stages that are being generated for, with an N of 6, 3 male and 3 female, assaying a small number of brain regions at this point. So these tend to be more cortically focused, but it's quite a broad data set. We, however, wanted to create some sort of an atlas structure where if your primary focus wasn't the neocortex, that you could find something interesting as well. And so we're creating this microarray data set where we're looking at 200, 300 LCM structures, again, in a developing human. And the data, we're using the same microarrays as we use for the adult human for doing those comparisons. We're also generating a series of high-resolution anatomical reference atlases for this project. So there's a mid-gestational, fetal, and adult digital reference atlases where we're generating high-resolution MRI and DTI. This is done with Bruce Fischel's lab at Harvard. We're doing fetal atlases with histological stains, and we're working on presenting all of this data in an integrated fashion. We're also doing large-scale cellular resolution in situ hybridization, again, complementing the work that we're doing in the adult human to look at a series of different genes across development and as well in the adult. And again, we are always limited here in the genes that we select. We try very hard to do things where we look at the same collection of genes in multiple species so that there can be interesting cross-species comparisons that can be made. This is how we are generating our LCM atlas for the fetal human brain. Again, we take samples, they're sectioned, they're put onto slides. Anatomists make determinations about what areas are to be cut. We collect the samples and we put them onto microarrays to get a quantitative readout. It's just shown here. You can see some of the variety of different sampling that we're doing in terms of collecting the data. How we create our reference atlases. This harkens back to a process that we used in generating the first mouse brain atlas at the Allen Institute. Hong-Wei Dong was involved in that process. This was effectively creating these data sets that have been stained with a variety of different histological stains. Once we scan these at high resolution, we actually print them out on these enormous prints. They get tacked to a wall, and then the anatomists go at it in terms of delineations, and we have a team of graphic artists that's going in and making these nice pretty pictures and the assignments there, and then the anatomists go back and QC them. So we're in mid-process of generating a series of atlases that will ultimately be web-based for this. As I mentioned, we've also been doing in situ hybridization on the fetal human brain, and there's something about developing brain where the data is just much cleaner than the adult. We haven't been able to quite figure out what that is. It could be that we're looking at transcription factors and other things, but in general, the data really looks dynamite in terms of this in situ hybridization platform. And so this is just an example of things where you can now start to do some cross-species comparison. You can see this is NPY turned on in the somatosensory cortex here, and just to show in the mouse, it actually doesn't, it's sort of quiet in 15.5, but if you go up just a few days, things are being rapidly turned on there. Just mention this again. This is more details of creation of the atlases. I'm going to fly through this. this is going to be a 20-year-old reference atlas, and this just shows the kind of quality that they'll be generating for that. And of course, as I mentioned, we're also doing imaging data in collaboration with Bruce Fischel's lab at Harvard, making anatomic assignments there, also trying to do some diffusion tensor imaging and pull all of that information together and integrate it with our atlases. All right, so just to summarize, in prenatal gene expression atlases, we've been generating in situ hybridization data, focusing on stage 3 and stage 4, which are these 15 to 25-week-old fetal samples, high-density coverage of developmentally important genes, again, going through and trying to find this subset of genes, which we think a lot of people will be interested in. It allows us for a nice comparison between developing mouse and human. Of course, we're creating these prenatal atlases, both the print version, the high-resolution histological atlases, as well as MR, and creating these prenatal microarray. And of course, don't forget, there's all of this RNA-seq, very, very deep information that's available from Yale and USC partners. So just to highlight, I always have to refresh this slide because we're constantly changing and generating new things. So the fresh numbers here that I just had our IT director, Paul, we've generated since our start in 2003, 3.2 million tissue sections. And I should state that as much as we try to automate things, those have all been done by hand by careful technicians. So I know there's probably many people out here who've felt like they've sectioned that many, but I guarantee you haven't. 850,000 microscope slides now, so we'll have to have some celebration when we hit 1 million. We've clearly blown through a petabyte of image data at this point, and that's actually probably more than 600 miles of data storage tape at this point. For perspective, the project that I'm going to talk to you about next, our connectivity atlas, we expect over a petabyte of data in a year's worth of production for that project. So again, things we couldn't have contemplated when we started our project in 2003. Nicely, technology moves forward. Multiple mashups of data, so people have mashed our data in various ways. Please mash it, we encourage it. It's nice to see translations, it's nice to see various ways in which people have pulled from the API and come up with creative ways of doing that. I'll tell you a little bit more about something that we hope to encourage that effort. And as I mentioned, through our human and our non-human primate work, we've generated 170 million gene expression measurements in both the HBA, the brain span development, and then in the non-human primate. So I'm going to switch gears here and talk about work that we're now doing to build on this concept of cell type from our mouse brain atlas work. So arguably the low-hanging fruit from our mouse brain atlas were genes which were selective in their expression within individual cell types, or something that closely approximated them in some way. Now, you won't get absolute selectivity, but you'll get certainly regional selectivity from genes that are being turned on. And so we hope to exploit this. We've done this a combination of ways. One is to create responder lines that are turning on in specific, Cree lines that are turning on within specific areas of interest. So mining the data, finding those genes, and then putting them, making the corresponding mouse line to be able to turn things on there. We've also been very interested in creating and expanding on the rapidly growing toolkit that's available for responding. And so this idea of giving you cell type specificity and building off of that in a way that I think is very exciting and sort of really points to the future of the field, certainly in mouse, of being able to speak from a common language, a common framework, to build on these resources. So we make we've made a whole series of different lines. This work was published a while ago. We've also created this toolkit, the Cree lines. We put all of the characterization data up there, so it goes through the whole brain, so there are no surprises about what you're getting. In addition, as soon as we generate the mice, we try to get a breeding pair off to JAX so that people can order them themselves. I think this is something we pride ourselves on, about getting these into the hands of many scientists. And I wanted an update. I couldn't get an update before this talk, but effectively, with these, we published a paper on these mouse lines in January 2010, but we're already distributing the lines prior to publication. And as of 9 months after this publication, 1,500 of these reported Cree mice had been distributed either by JAX or by the Allan Institute. And we've continued to do that. It's continued to grow. Share your mice. So we're now going to take advantage of that for this first project we'll talk about, the connectivity atlas, but also our future efforts at the Institute. So this project is designed to create a wiring diagram of the mouse brain. This is not meant to be exhaustive, but rather to cover 300 to 500 brain regions using these cell type specific reporters, these Cree mice, combined with Cree-dependent viruses. We're also doing some standard fluorescent tracing as well to survey the brain. Followed up by high-throughput fluorescence imaging, ultimately capturing this data and creating these connectivity models. So it's early stages. We're just now in the process of piloting the work, but actually things are going quite well. We started out with a prototype system. This is the tissue site system from a company called Tissue Vision. Effectively, there's a fixed brain. It's been, there's fluorescence tags that are Cree-dependent viruses turning on fluorescence tags within individual brain areas. They're labeling individual connections within the brain in a cell type specific way. And the brain is then fixed and put in here. And this is effectively a microtome combined with 2-photon imaging. So the 2-photon imaging is capturing, and then it cuts, and it collects, and it cuts, and it collects. And that ultimately gives us a nice three-dimensional structure that doesn't have the limitations of the things that we did with our slide-based methods where you get these distortions and other things. It actually gives you, because you're doing this sort of block-based imaging, a nice full three-dimensional model. And this is the Thigh-1 mouse. Many people have used the Thigh-1 mouse, but it just shows you the kind of data set that you can get out of here. In a second, I think it's going to fly through here. It's not perfect. You can definitely still see some things here, but it's a pretty extraordinary data set to generate, and it does that in about 3 hours. Again, it's really stunning, and when we're starting to do these tracing studies and be able to collect this data, it's really nice. We're gearing up for having 6 of these machines going as we're ramping up into high throughput over the next year. So watch this space. We're going to release our first data set in November. Lots more to come in a very systematic way that hopefully people have come to expect from the Allan Institute to generate this data set. So just to go back to the website a little bit, please continue to give us feedback. The only way that we can improve our data, the only way that we can hear from you is when you give us feedback in a variety of different fora. Certainly on the website is the best place for us to capture it and appropriately track it. We're trying to get out more into the community for doing training sessions. So we now have scheduled training sessions at major neuroscience conferences, as well as some regional sessions where our team is going into a variety of different areas: Texas, RTP, Bay Area, LA, San Diego, Boston, NIH, DC, New York City, Toronto, and certainly let us know if there's a lot of interest at your institution and you feel like you'd like to host a training session. Please let us know. We'd be delighted to put you in the queue. I mentioned briefly about a desire to get more mashups and other things. We're going to be likely hosting a hackathon in Seattle next summer to get people to come in and really create, hopefully, unique, interesting tools that sort of build off the data that we've generated. Okay, good. Last 5 minutes, I'm just going to cover a little bit about future scope of the Institute. Again, this is early stages. Christoph Cook just joined us in April of this year, but Christoph joined us with the thought that we were going to move from cell types and connections into neural computation, behavior circuits, ultimately moving in this direction. So we're contemplating something very big in that space. We're very excited about it. And this is really about trying to understand neural coding. We decided we're going to try to do this in the mouse brain. We're going to focus on the visual system and do the various things here, and I'll tell you a little bit about how we're envisioning doing that. So this is described in very great detail all these genetically identified cell classes. So this is connectivity atlas leading into this. Recording, so there's going to be an active component of calcium imaging and electrophysiology, interfering with using these latest and greatest optogenetic tools for doing this. But an important thing is on theory and modeling, so modeling and understanding. So we hope to have this productive loop of modeling, theory, and wet lab all coming together. Christoph said this the other day. We thought it was kind of humorous, perfect vision in 2020. The idea here is really to take things to scale. We call these sort of high-throughput brain observatories, these areas we're going to create, again, in our way of doing high throughput, a series of these large-scale efforts. I shouldn't underestimate or downplay the fact we're going to be doing some level of EM. We need to integrate all of these together. Again, trying to get these under one roof is going to be a challenge, but we think that the iterations therein are going to be really important for for tackling some of these big challenges. And we think that the scale and integration, again, always offer interesting opportunities for establishing field standards. And so this is something that's very nascent at this point. It's in the planning stages. We're hoping to get sort of final approval to move forward. So again, I would encourage you to watch this space. We're going to be building out teams. If you have expertise in any one of these areas and you're looking for something that's going to be, I think, on a larger stage and you really enjoy the sort of team science work, I encourage people to keep a close eye on what we're doing here over the next several months and let us know if you're interested. So broad acknowledgement here. I take credit for none of the work that I presented today. It was all done by a tremendous number of great people, both at the Allen Institute and others. Of course, much of it was funded by Paul Allen and Jody Allen, but there's certainly the NIH has funded a lot of our developing human and non-human primate work as well. Just briefly, I'd like to say to Ted Jones, he sat on an advisory council for the Human Brain Atlas for several years. We certainly are going to miss him. Spirited debates, always fun to see him mix it up with folks, and we're really, really going to miss him. Lots of other acknowledgements. Huge number of people help us. Many are probably sitting in this room. And I can never do justice to it all, so I always say to go to the website to get more information. And with that, I'll conclude.